Meet Raymond, a famous model from Indiana who has worked with many popular clothing lines like Salty Crew, and yeah, that's actually about it. But, anyways, Raymond has somehow acquired a lot of money from his fashion career, and he can buy almost anything. And what do you do with a whole lot of money? Well, of course, you clone your bird. Yep, that's right. Raymond here is going to clone his beloved bird so he can be with him forever. While I might be able to clone this majestic duck myself with a simple Control C, Control V, in real life, Raymond would have to go through a much more scientific process, specifically somatic cell nuclear transfer, or SCNT, for all those lazy scientists out there who like to abbreviate everything. In order to talk about SCNT, or somatic cell nuclear transfer, I'm actually going to be talking about a sheep that probably many of you have heard about. Dolly the sheep was created in 1996 and became the first clone of an actual breathing mammal. So I'm sorry, Raymond, but your bird will not become the first living clone. But how was this sheep cloned using this somatic cell nuclear transfer? This was the stuff of science fiction which has only now become reality after over a century of continuous hypothesis. Well, first, a somatic epithelial cell was isolated from the sheep wanting to be cloned. This cell could be taken from the individual itself or could be taken in culture or from frozen tissue. Next, a pipette or very thin needle was used to puncture a hole into the cell and remove the nucleus with all of its DNA from the cell. After this, a cell was removed from an oocyte undergoing metaphase 2 of meiosis, which is important since it is right before anaphase and cleavage of the cell during cytokinesis. This cell was then enucleated. The nucleus was removed, and another needle and pipette was used to poke a hole in the enucleated oocyte and insert the somatic nucleus inside. Electric fusion is then used to combine the egg cell with the somatic nucleus. This cell is then stimulated to undergo embryonic development, many of which fail. After a series of mitotic cell divisions, the oocyte, which has the nucleus and therefore the DNA of the animal wanting to be cloned, becomes a blastocyte or pre-implementation embryo. The blastocyte is then inserted into the uterus of a surrogate mother to undergo full embryonic development and hopefully birth a clone of the nucleus donor. Before we begin looking into this further, though, we should talk about the somatic cell that the nucleus came from. August Weismann in 1855 discussed a largely prominent hypothesis that the cell was used, which would contain the DNA from the animal wanting to be cloned, had to be in an undifferentiated cell. This was justified by the theory that during development, either part of the genome of a cell is lost or turned off, allowing for the cell's fate to be decided. You see, each somatic cell in your body, from your skin to your eye cell, should have identical nucleuses with the same DNA, 46 chromosomes and genes that code for you. But each cell can only access certain cells. So, if you're a skin cell, you might have specific genes regulating melanocytes production or transportation, and proteins that make up the skin's texture, but not proteins making the eye. Since each cell had access to only a specific gene, many thought, especially due to not having proper tests, that these differentiated cells could not be used to create a whole living being, and could only be cloned to create other skin cells or other cells of its somatic cell. When Dolly the sheep was cloned, an epithelial somatic cell of either the skin or surrounding of organs was used for the cloning nucleus, and not a differentiated cell. The previous problem proposed by Weismann was solved by entering the somatic cell into a G0 state of starvation, a state replicating prevalent stages in embryonic mitosis and meiosis development. This is called serum starvation, and this reminiscent state allowed all of the genes to be accessed, and when it worked, it demonstrated that inactivated genes during tissue differentiation could be reactivated through a concept called nuclear programming, which has many methods allowing a differentiated cell to revert back to its totipotent stage. An interesting point to investigate is that there will be some phenotypic and even genotypic differences in the two mammals. For one, mutations in larger epigenetic environmental methylation and acetylation can occur. Also, mitochondria, which are outside the nucleus, also have their DNA, which is passed down from the mother. Due to this, since only the nucleus is taken, The, the DNA in the mitochondria is not the same, even though all of the main outside genes are from the nucleus. Cloning is an interesting topic which, while surrounding many achievements, as it could lead to the cloning of mammals with modified proteins that could produce certain pharmaceutical drugs, could clone resistant animals, and could bring back extinct species, it is also controversial, as it brings up the question of, are humans really ready to play the role of God? Thank you, and see you soon.